So for this last lecture in this session, I want to move on from the grid cells and just look briefly at some of the other cell types that have also been discovered in the hippocampal region. So the place cells and the grid cells and the head direction cells have been really notable for the very clear signal that they have and the way that we intuitively understand that these are components of a map, the compass and the scale and all of that. But when we look in these regions of the brain that communicate with hippocampus, we also see a bunch of other types of cells. And we're slowly beginning to piece together a picture of how they're all interacting. And it turns out to be quite a sort of a complex network. So, for example, we've known for a long time that in amongst the cells that we've been talking about, which are called principal cells, so these are cells that have long axons that stretch from the region where the cell is to some other region. So, for example, from entorhinal cortex to hippocampus. And principal cells are excitatory, so they use glutamate as their neurotransmitter, and their action is to stimulate neurons that are downstream in the chain. But mingled in with all of these principal neurons are many, many other types of neurons called interneurons. And these are neurons that act locally. So instead of having these long axons that reach into other regions, they have short axons that reach locally and often quite branched. And these neurons are inhibitory. So they use GABA or glycine, which are inhibitory neurotransmitters that cause a cell that they contact to, instead of depolarizing to become more excited, it will hyperpolarize and become less excited and less excitable. So they're sort of dampening down activity. And we used to think that interneurons were just there to kind of regulate the background level of activity and make sure it didn't, things didn't become too excited. But there are so many types of interneurons, and this is a summary that was put together by Klausberger and Smogy a few years ago, and it shows us that there are many, many different types of interneurons, even just in the hippocampal region. They all have different shapes, and they have different extents of reach. And also it turns out that they have different temporal firing characteristics. So if you look at these different neurons in terms of the place that they fire in the theta cycle, so the phase of theta, then these each neuronal subtype has a different preferred phase of theta that it likes to fire on. So we're really building up a picture of these interneurons as being like an orchestra, sort of many different types of neurons, and they're collectively shaping activity. And we don't even begin to understand yet exactly what they're doing. So that's a big question to be solved in future. Another type of activity that we quickly started to realize was very prevalent in these regions are so-called conjunctive neurons. And these are neurons that have some mixture of the properties that we've been talking about. So we've been talking about place fields as things that are produced pretty much in isolation, and head direction cell tuning curve is a fairly pure signal, and a grid is a pure signal. But in entorhinal cortex, for example, there are many cells that have conjunctive properties. So, for example, this paper by Sargolini et al. Was the first to show that in the medial entorhinal cortex, where the grid cells are, in the deeper layers, so if you go down into the layer 5 and 6 of the entorhinal area, you see cells that look like grid cells, but they only fire when the rat is also facing in a particular direction, for example. So they are conjunctive grid bidirection cells. And there are some cells that are theta modulated, so they are combined direction by theta or place by theta and so on. So these conjunctive cells that seem to combine properties may be steps in a processing hierarchy where information is being kind of put together, if you like. And we're still trying to work out quite why that's happening. Conjunctive cells are not just found in medial entorhinal cortex, but also they've been found in a number of other brain areas. For example, you can see the border bidirection cells, grid bidirection cells, place bidirection cells, and retrosplenial cortex, and so on. So conjunctive coding is looking like it might be quite a general property of brain processing. Another important type of neuron that was rapidly discovered in medial entorhinal cortex is so-called border cells. These were actually predicted some years previously and first reported in subiculum. So border cells are cells that are sensitive to the border of the environment that's located in a particular direction from the rat. So, for example, there may be a border cell that responds when the rat is near the north wall of the box. And if you remember back to the boundary vector cell of Burgess and colleagues, they predicted that there would be cells that fire at particular distances from walls located at particular directions from the rat. And so that's how a place cell is able to know how to position its field relative to the west and the south wall, for example. So that was the idea. And so when these cells were discovered in subiculum and then later reported in the medial entorhinal cortex, it began to seem like maybe these were the cells that had been postulated. So we now know that place cells and grid cells are very sensitive to the boundaries of the environment. We've seen how stretching the environment can pull apart place fields and stretch grids, and so on and so on. So it may be, in fact is likely, that these border cells are the cells that are mediating the influence of the borders on the other types of cells. Border cells have now been found in subiculum, entorhinal cortex, and also this little brain region called the claustrum, where cells have been found that respond to borders not in a particular direction but all the way around the perimeter, so perimeter cells. So there are many regions in the brain that are kind of interested in the borders of the environment. Now, another type of neuron that's been found to have a relatively pure signal, and again in medial entorhinal cortex, it seems to be kind of a collection place for many different types of signals. And so we've known for quite some time that the spatial cells need to have some kind of information about how fast the animal is running, because, for example, a place cell will always produce a field of the same size no matter how fast the rat is running. So it needs to kind of know how fast the rat is running in order to compensate for the shorter time it takes. So there needs to be some kind of speed input. And in this experiment in the Moser lab, what they did was control the speed of rats on a linear track that had a motorized car and a treadmill, so that the rat could be forced to run at particular speeds with this car that pushed it along, and the track was actually quite long. It was 4 meters, so the rat could be brought to quite a high speed and then down to a low speed and so on. And what they found is that there are neurons whose firing rate tracks the speed of the rat really accurately, and you can see that in the bottom traces, a linear relationship between firing rate and speed of the rat. And in the far right trace, you can see when there's a sudden drop in the speed, the firing of the cell drops dramatically as well. So these cells are very responsive to speed. So these may be partly the way in which grid cells and place cells are able to keep the spatial, the metric characteristics of their spatial firing intact, independent of the speed that the animal is running. Now, some other types of neurons have also been found that that seem to suggest that this system is collecting information from a variety of different spatially relevant sources, and landmarks and objects, as we've seen, are one type of spatially relevant source of information. So this interesting set of studies looked at what happens if there's an object in the arena, and it turns out that there are some medial entorhinal neurons that are sensitive to objects and will fire at a particular distance and direction from an object. And if the object is moved around, then the cell will shift its firing to follow the object. So it's not anchored by the boundaries like the grid cells or the place cells and so on. Even more interesting, a subclass of neuron was found that will fire when an object is placed in the environment, and will continue to fire when the object is removed. So it'll remember, as it were, that there had been an object there. And then if the object is replaced in a different location, this neuron will acquire a new firing field around that new location of the object. And again, when you take the object away, that new field will persist. And so over the course of this trial, you can see here that this cell has built up a whole collection of firing fields corresponding to previous locations of the object. So these are really particularly interesting cells because these are, if you like, a memory for the previous location of objects. And it may explain why it is that if you take an object away, when an animal goes back into that environment, it'll sniff around the place where the object had been. So the animal clearly remembers that there had been an object there, and it may be that these object trace cells are the way in which it's able to do that. So that's just a few of the new subtypes of neurons that we've discovered since the place cells were first discovered many years ago. You can see that it's really a complicated collection, and so the brain has a lot of different subtypes of information, all pertaining to knowing what is where in the environment. And so this is sort of telling us that the neuronal coding of spatially relevant information is somewhat specialized. So we have these neurons that have these specific coding properties, and then there are also these other neurons with the conjunctive coding properties that are putting subcategories of information together. So there's a very, very heterogeneous kind of confluence of information going on. And it's a sort of a very mosaic kind of picture that we're building up. And we still think that we've got quite a way to go before we've uncovered all of the types of information that are encoded. And also, we think that these are distributed in quite a few places in the brain that we haven't yet discovered yet. So this spatial map is really a very distributed network. So in the next session, we're going to move on from space to something that's only recently become apparent about the hippocampus, which is its involvement not just in space but also in time.